有好多同學問呢啲片喺邊度揾啊？其實係翻留言區，我一嘢拍晒落去，大家撳翻入去呢，有得睇噶啦。
我要晒，有得啫。
之後呢，梁 Sir 會拍一段片呢，就會有埋筆記啦，去教大家呢嗰個隔離唔同嘅方法。書本冇咁多噶，但係過往考嘅題目呢，係出晒唔同類型嘅隔離方式嘅。2022年第10條題目咧係橫跨咗3個課題嘅。第一就係分類啦，第二就係演化，而第三咧又係科學探究啦。咁我哋嚟睇一睇題目咯。蘭花植物嘅其中一個屬名咧就係 Hibernaria， 亦即係玉鳳花屬啦。佢嘅花朵咧顏色暗淡而有香味嘅，係可以吸引到啲蛾啦喺夜間咧去傳粉嘅。而呢個屬入面咧有一種特殊嘅物種，就叫 H. roticula 紅唇花。顧名思義啦，佢嘅花朵咧就呈現紅色，但係啦就缺乏一啲可以察覺嘅香味。已知道啦，夜間咧係冇昆蟲為呢個紅唇花咧去傳粉嘅，但係昆蟲物種 A 咧就會喺日間採食佢嘅花蜜啦。咁都唔難理解嘅。嗱，雖然佢係紅色啫，但係夜晚黑未必有人見到佢，佢又缺乏嗰種香味嘅話啦，咁夜間自不然就冇乜昆蟲咧係為呢個紅唇花去傳粉啦。但係朝頭早嘅時間啦，咁我由於佢係紅當當啊嘛，咁啲昆蟲物種 A 咧就見到佢啦，咁就會咧採食佢嘅花蜜咯。咁而家 Part A 咧就入題目啦。以下呢個二叉式檢索表或者平時我哋講嘅二分鍵啦，就可以用嚟鑑定到昆蟲物種 A 咧係屬於邊一個組別。咁我哋有組別 PQRS 啦。咁而家啦，我哋就睇翻呢一幅圖咯，顯示咗物種 A 咧喺靜止時候嘅外觀。咁我哋就利用翻咧呢個二叉式檢索表咧，就寫出一個鑑定嘅次序啦。呢個題目啦，自不然就考我哋用翻二分鍵去揾翻一款生物啦。咁所以啦，嗱睇翻過往有一段片咧，教大家點樣去分唔同類型嘅生物嘅。好，咁跟住啦，睇翻題目咧，佢要我哋寫出鑑定次序啊嘛。嗱，寫出鑑定次序咧有2個方法，第一個咧就係箭嘴，第二個就係逗號。嗱，冇咗呢2樣嘢咧就冇意思啊，hyphen都係冇用噶吓，你有個指向性或者有個次序性嘅。咁而家啦，先去睇下咯。嗱，有翼啦，係嘛？1A 啊，有翼咯，有翼之後就去2啦，去2之後咧就睇下啦，佢觸角係長過個頭啊，定係短過個頭咧？嗱，個頭就喺呢度，觸角就喺呢度，咁自不然都係長過個頭啦，就落3咯。咁即係話啦，頭2 part 咧就係 1A 箭嘴 2A 咯。咁去到3A 3B 咩意思咧？咩叫做昆蟲靜止時翅膀平放，又或者昆蟲靜止時翅膀垂直咧？嗱，咁而家呢個當然係垂直啦。咩叫平放咧？好似呢類型咧就叫做靜止時翅膀平放啦。咁而家我哋睇翻呢個昆蟲物種 A 咧，自不然就係靜止時翅膀豎直啦。咁所以就係 1A 箭嘴 2A 箭嘴 3B， 所以佢就係組別 S 嘅昆蟲咯。咁過往啦都有唔少嘅題目咧係考我哋用呢個二分鍵咧去做嘅。咁啊快手睇下片溫下書啦。然後啦就去到 Part B 咯。喺呢個個案當中咧 H. roticula 紅唇花咧就由其他嘅Hibernaria species 玉種花屬嘅物種咧去進化而嚟嘅。題目就要我哋啦，試提出唔同昆蟲嘅傳粉者點樣促進到呢個 H. roticula 紅唇花嘅物種形成呢？嗱，今次嘅重點咧係擺咗喺昆蟲嘅傳粉者嗰處。諗下啦，玉鳳花花屬咧本身就係暗淡，但係有香味嘅，咁就會吸引到啲蛾啦喺夜晚黑幫佢傳花粉。但係呢個紅唇花唔同喎，夜晚黑冇人採佢嘅，係朝頭早嘅時候先有呢啲昆蟲物種 A， 類似咧係啲蝴蝶啦，係咪？就幫佢去傳花粉喎。咁點樣促進到個物種形成呢？嗱，咁呢條題目啦，唔使審啦，梗係考翻我哋有關於物種形成嘅機制啦。同時啦，亦都考下我哋啦，基因變異嘅來源究竟係點樣？嗱，因為當初咧，大家都係屬於玉鳳花屬嘅祖先嚟嘅。咁但係點解啦？到頭來有啲紅唇花就出現咗呢？咁當中啦，有啲重要嘅字眼咧，都提埋大家先啦。合子前嘅隔離啦，即係話啦，唔同嘅生物咧，佢係連交配嘅機會都冇啊。 第二啦就係基因變異啦、突變啦、自然選擇啦。咁而家啦，我哋摸順翻個脈絡咯，究竟有啲咩重要嘅概念呢？我哋係要知道翻啦。第一個呢，就係啲共同祖先，佢哋係點樣受到隔離嘅呢？一開始啦就有呢個蘭花植物嘅玉鳳花屬啦，咁大家都共同祖先嚟嘅。只不過啦，突然之間呢，有啲突變就產生咗啦，有啲蘭花呢，就變咗做紅色，但係冇味喎。其他咧就仍然係暗淡而有香味喎。咁呢個隔離啦係點樣導致嘅呢？就係喺呢一批嘅玉鳳花屬嘅共同祖先當中咧，呢個暗淡而有香味嘅花，同埋呢個呈現紅色但係缺乏可以察覺香味嘅花咧，就唔能夠再做到一個異花傳粉啦。嗱，呢個好緊要，係因為題目係講到明係一個昆蟲嘅傳粉者嘅角色啊嘛。因為啦，佢哋吸引緊嘅已經係不同嘅昆蟲傳粉者啦。暗淡有香味嘅花，夜晚黑就靠啲蛾咧去傳花粉嘅。但係紅唇花就要靠朝頭早嘅昆蟲物種 A， 似係一個蝴蝶啦去傳花粉咯。仲要啦，就令到呢兩種花咧隔離開啦，亦都各自有各自嘅演化咯。而最緊要，由於佢哋做唔到一個異花傳粉啊嘛，自不然啦，喺呢兩個群落當中咧，都唔會有一個基因嘅流動咯。然後啦，就去到自然選擇咯。究竟啦，各自嘅物種係點樣選擇呢？因為你諗下啦，佢哋已經係唔能夠再做異花傳粉啦，各有各嘅突變，各有各嘅基因變異，各有各受到自己嘅自然選擇。所以你睇翻書本，佢答案係非常之相近噶啦。好多代之後，個基因變異就會累積起上嚟，累積到一刻，佢哋唔能夠再交配，產生一個有繁殖能力嘅後代為止，佢哋就變成兩個完全獨立嘅物種啦。其中一個咧受到昆蟲物種 A 去做傳粉嘅就係 H. reticula 紅唇花啦。咁過往咧都有唔少題目咧都係講物種形成嘅，有 MC 有長題目，你睇翻嗰個答法，其實同我哋書本係非常相似，不過個隔離方式好唔同啦。之後咧，梁 Sir 會拍一段片咧，就會有埋筆記啦，去教大家咧嗰個隔離唔同嘅方法。書本冇咁多噶，但係過往考嘅題目咧係出晒唔同類型嘅隔離方式嘅。咁下次段片咧就敬請留意啦噃。好，跟住啦就去到 Part C 啦。為咗揾出翻昆蟲物種 A 啦，究竟啦佢係被紅唇花嘅外觀啦，定係香氣所吸引呢？啲研究人員啦就設計咗一個探究啦。咁如下圖所示咯。咁我哋啦有設定一啦，就係一個視覺裝置，透明但係冇窿窿嘅容器。另一個咧就係對照裝置嚟嘅啫，透明亦都係冇窿窿，不過入面冇咗呢個紅唇花啦。跟住啦，設定二咧就係嗅覺裝置，佢係唔透明嘅容器，不過有窿窿嘅。同樣地啦，都係對照裝置啦，就係冇咗棵植物咧，亦都係唔透明，亦都係有窿窿。咁當中你梗係要明白到啦，對照裝置冇花嘅，就係想我哋咧去比較結果嘅時候，可以知道係因為真係朵花嘅問題，因為佢朵花喺對照裝置唔存在啊嘛。喂，咁啊真係嗱，都有一次嘅探訪嘅，咁但係呢度咧43次咯。咦，點解咁有分別嘅咧？就係因為朵花嘅存在啦。咁設定3咧，陣間再講嘅，因為咧喺暫時第一部分、第二部分咧係未問到嘅。咁而家我哋睇翻整個裝置嘅詳細嘅內容啦。每一個嘅設定咧，喺兩個裝置放喺一個空曠嘅地方啦 ，open area 啦，兩者之間距離60 cm， 維持1個小時，然後再將兩個裝置位置互換，又再放1個小時。喺呢2個小時入面啦，物種 A 嘅個體飛近任何一個裝置10 cm 範圍以內咧，就即視為1次嘅探訪噶啦。而物種 A 對每個裝置嘅探訪次數咧，亦都如圖所示啦。喺設定1咧，視覺裝置有43次嘅探訪，對照裝置1次探訪。去到設定2咧，嗅覺裝置同埋對照裝置咧都係得1次探訪嘅啫。咁而家第一部分咧就問我哋啦，為咗確保實驗係公平嘅測試，有必要咧將兩個裝置嘅位置互換嘅，試加以解釋啦。嗱，頭先有講到噶嘛，佢大家咧放咗一個空曠地方，相距60厘米嘅。咁但係啦，放完第一個鐘之後咧，兩個嘢就乾坤大挪移咧， 就倒轉位咯。啊，點解要咁樣做呢？其實條題目咧就考翻我哋對於公平測試啦、對照裝置啦嘅重要性。對照裝置就係欠缺咗要探究嘅因素，今次要探究因素就係棵植物啊嘛，所以佢咪冇咗啦，係咪？所以啦，佢係可以確保到實驗結果咧係由探究嘅因素所造成嘅，亦即係話呢棵植物有冇存在到咯。而第二部分咧就問我哋有關於控制變項嘅概念啦。咁有啲咩控制變項啊？例如啦，兩個裝置佢哋相距距離係要60 cm 啦，係咪？就唔好咧頭一個鐘咧就60 cm，第二個鐘咧就放30 cm。咁成個實驗咁咪唔準確咯，直頭做錯嘢添啦，係咪？即係整個容器嘅大細啦，又或者啦個空曠位置咧都有控制變項嘅喎。設定1咧就擺喺空曠地方嘅，擺喺個公園度。而設定2咧，我都係擺喺空曠地方，不過我擺喺個地盤度。嗱，兩個都好空曠嘅，兩 Sir 啱啦啩？你唔好玩啦，一個擺喺地盤，一個擺喺公園，已經唔係一個控制嘅變數啦，應該一樣噶嘛，係咪？咁而家呢道題目啦，就係問緊點解擺完1個鐘之後，佢要左右倒轉乾坤大挪移呢？其實就係關我哋一個隱藏嘅 CV 事啦，就係嗰個位置問題啦。我哋係為咗肯定得到兩個裝置所獲得嘅數據呢，係唔會受佢嘅位置所影響。呢個永遠擺左邊，另一個永遠擺右邊，人哋咪會挑戰你咯。或者啦，嗰個昆蟲物種 A 呢，佢去揀選個裝置呢，我哋話俾人哋聽，唔關佢位置事嘅。佢總之見到朵花或者聞到朵花，佢就會過去噶啦。你就算有左右調位呢，佢都會過到去嘅。啱啦，就係為咗消除翻喺呢兩個位置所帶嚟嘅一啲唔同嘅物理因素嘅影響，例如溫度，例如光照，例如風速。OK， 諸如此類嗱，所以咧你要俾個例子我，咁呢個都係關乎於個位置事嘅，而頭3個咧都係關嗰棵植物嘅位置事嘅。到第4個嘅原因咧，就係有關於嗰個昆蟲事啦，係我哋想減低翻呢隻昆蟲行為嘅慣性啦 ，behavioral inertia 行為慣性啦。例如啦，如果呢隻昆蟲咧，原來佢嘅飛行時間係好短嘅啫，佢耐力不足噶，咁所以啦，你擺近啲咧。佢咪過嚟咯，你擺遠啲，佢唔過嚟噶啦。所以啦，我哋將呢啲裝置咧左右倒轉乾坤大挪移啦。我哋就想去睇下，嗱，你真係受到花吸引噶嘛？你受到花吸引，咁即係話你想去，我無論擺幾遠或者擺幾近，你都應該會去嘅。就想去減少翻，喂，究竟咧你呢隻物種咧，究竟飛行時間長唔長噶？你會唔會太懶啊？嗱，佢咧淨係採近嗰個花嘅啫。遠啲我唔睬噶啦，所以你行遠啲咧我就唔過嚟噶啦。所以嗱，我擺完1個鐘之後，咪將佢左右倒轉咯。嗱，頭先又遠啫，而家近啦啩，過唔過嚟啊？原來啦，無論擺遠定擺近，唔過嚟就係唔過嚟。又或者啦，你係要過嚟嘅話，擺到老遠你都會過嚟。咁過往例幾多題目咧講埋啦，實驗裝置同埋對照裝置，你識唔識得分呢？用10年嘅 past paper 去教翻你咯。有好多同學問呢啲片喺邊度揾啊？其實係翻留言區，我一嘢拍晒落去，大家撳翻入去呢，有得睇噶啦。跟住啦，去到第2部分啦，就叫我哋參考翻探究嘅目的啦。究竟啦，由設定1同埋設定2啦，我哋能夠得出啲咩嘅結論呢？呢個題目啦，就考翻我哋嘅傳視技能啦，就係點樣獲得結論啦。同一時間啦，就係要我哋學識翻去比較裝置1同裝置2，或者佢哋嗰個視覺裝置啊、嗅覺裝置同個對照裝置之間啊，佢嗰個探訪次數嘅分別。然後用直線抽擊答題法去得出結論咯。咁題目呢講咗俾我哋聽噶嘛，參考探究目的。咁我哋梗係要知道翻探究目的啦。不厭其煩啦，講多一次啦。為咗揾出翻昆蟲物種 A 係受到紅唇花嘅外觀，又抑或係香氣所吸引。所以啦，研究人員咧就設計咗呢一個探究咯。咁我哋啦就嚟睇下設定1先啦。設定1我哋會發現啦。視覺裝置對比起對照裝置咧，咦，43次嘅探訪，遠高於對照裝置嘅探訪數目，所以啦，有翻呢一個比較先啦，係嘛？咁跟住啦，就到結論啦，直線抽擊答題法啊嘛，你望到啦，視覺裝置探訪數次多過對照裝置咁多，似乎嗰個昆蟲物種 A 咧係受到朵花嘅外表外觀所影響。喂，咁關唔關香味事啊？睇埋設定2啦。設定2就係嗅覺裝置對照裝置，兩個都係得1次探訪啫，直頭一樣。 咁自不然啦，原來有香味都冇咩優勢啫。嗱，喺設定一、設定二呢，我哋就有呢兩個嘅結論咯。一年都總會有一啲噶，唔係 MC 呢，其實就係長題目嚟噶啦。咁所以啦，大家咧睇下片、溫下書，學翻晒啲實驗技巧咯。好，跟住啦，去到第三部分啦，介紹埋設定三先。設定三咧就係有一個裝置就叫視覺和嗅覺裝置，佢咧就係一個透明嘅容器啦，亦都係有窿嘅。對照裝置咧，亦都係透明，亦都係有窿，不過就冇植物嘅。咁得出嚟咧就係48次嘅探訪對2次嘅探訪啦。好，咁我哋睇一睇題目咯。咁啊，阿 N 同阿 Ken 咧就比較咗3個設定嘅結果啦，佢哋有唔同嘅見解啦。嗱，當初咧頭先喺第二部分咧，設定一、設定二咧，我哋會知道嘅昆蟲物種 A 咧，似係受到個紅唇花嘅外表咧去吸引嘅，同個香味咧好似冇乜拉間嘅。不過啦，阿 N 同阿 Ken 咧，佢哋有唔同嘅見解。阿 N 就認為啦，設定3咧，係利用呢個視覺和嗅覺裝置，即係話一個透明兼且有窿嘅容器呢。咦，佢吸引咗48次嘅昆蟲嘅探訪喎，會唔會係反映咗一個協同嘅效應啊？但係阿 Ken 就話啦，唔關事嘅，其實呢係隨機差異嚟嘅啫。得啦，我哋為咗驗證阿 N 嘅見解呢，佢哋就決定進行另一項包含2組設定嘅探究去做比較啦。整個實驗咧就係考我哋點樣去建構一個對照裝置啦。記緊翻啦，對照裝置咧就同佢嘅實驗裝置就只有一個差別，呢個差別就係我哋嘅探究嘅因素，就係我哋嘅獨立變項，亦即係 IV 啦。好咯，我哋又要睇下個實驗嘅目的啦。我哋係為咗驗證阿 N 嘅睇法，究竟嗅覺同視覺吸引有冇一個協同效應？所以啦，我哋陣間要做嘅咧，就係將呢一個視覺同埋嗅覺嘅裝置同視覺同嗅覺佢哋各自獨立地比較。我哋要比較就係，咦，視覺嗅覺有協同嘅效應，我哋咪就利用呢一個視覺嗅覺嘅裝置同翻各自嘅視覺同埋各自嘅嗅覺嘅裝置做個 battle 咯。所以啦，我哋呢兩個裝置咧非常簡單啦。第一個裝置咧就係有植物、透明容器，不過冇窿窿，亦即係我哋嘅視覺裝置，係咪？視覺嗅覺裝置對視覺裝置，就睇下啦，究竟嗰個差別係咪真係咁大？你話協同效應啊嘛，阿 N。而設定5咧就係有植物，但係唔透明嘅容器。而有窿窿，亦即係我哋嘅嗅覺裝置啦，亦即係將視覺嗅覺裝置同嗅覺裝置去比較啦。嗱，三隻都係嗰句啦。阿 N， 你話協同效應㗎嘛？咁即係話啦，視覺嗅覺裝置咧應該係贏嗅覺裝置好多嘅。咁所以啦，如果視覺裝置得出嘅數據係同視覺和嗅覺裝置得出嘅數據係差無幾嘅，亦即係話啦，阿 N 所講嘅協同效應咧，就似乎站不住腳啦。但係如果我哋下次再做多一次啦，咦？視覺和嗅覺裝置佢有100次嘅探訪喎，但係視覺裝置原來都係得43次嘅探訪嘅話呢？咦？咁似乎阿 N 又啱啱地喎。OK，嗱，咁其實今次呢，設定4同設定5嘅答案呢係可以相反嘅。總之其中一個設定呢，就係呢個視覺嗅覺嘅裝置同一個視覺裝置嘅比較。另一個裝置咧就係同嗅覺裝置比較，咁你即使上下答案倒轉都冇乜所謂。對於實驗嘅題目咧，大家嘅思路咧要非常之清晰。而呢條題目啦，有啲咩嘅題型變奏咧，又教下大家啦。下次出題目啦，就可以問下你啦。參照翻我哋22年1B 第5條，我就問下你啦。咦，有咩辦法去改良下呢個實驗，去增加翻呢個探究嘅可靠程度啊？試下喺留言區留低嘅答案。好，跟住啦，去到1點出發啦。呢條題目咧就講開花植物嘅，考我哋2樣嘢啦。第一就係分類，第二就係演化。先講分類啦。今次嘅花咧主要講嘅係蟲梅花，開花植物都會有風梅花噶嘛。兩者有咩比較呢？有咩佢哋嘅優勢，或者有佢哋嘅問題需要面對呢？同一時間啦，題目亦都考我哋用2分鍵喎。咁提翻大家啦。2分鍵咧就係利用翻嗰隻生物嘅外在特徵咧去做個分別嘅。 然後啦就去到演化咯，今次題目問物種形成啦，下次又可以問翻演化樹咯。過往都有唔同 MC 去問演化樹長題目呢，其實我哋都係考過噶。然後啦就去到物種形成，自不然講隔離啦、基因變異啦、自然選擇啦。過往咧都拍咗好多片噶啦，親緣關係啊、演化樹啊、演化嘅邏輯啊，有好多片噶，快手睇下片溫下書啦。而今次嘅題目再結合嘅概念就係考埋科學探究啦，我哋要探究咧，就係呢個昆蟲傳粉者啊，係受到朵花嘅外觀啦，定係香氣啦去吸引。題目啦就問我哋有關於對照裝置嘅概念啦、CV 嘅概念啦，過往都拍咗 IBDV CV 啦。對照裝置啦、實驗裝置嘅片啦，咁啊快啲溫下書啦。今次個題目咧就要我哋得出結論嘅。咁其實啦，下次又可以問翻假説啦，或者問下我哋嘅預測啦。例如啦，今次阿 N 咧佢就假説啊嘛，個紅唇花咧對啲昆蟲物種 A 咧就有一個視覺嗅覺嘅協同效應。我就當阿 N 你係啱嘅話啦，咁我哋個結果預測會係如何咧？我叫你預測完個結果，我再俾個真嘅結果你，你咪得出結論咯，係咪？好，咁大家留意下一段片啦，又有筆記咧就送俾大家啦噃。Question 10 is about 3 topics. The first one is about diversity, classification. Second one is the evolution. And the third one is about the scientific investigation. Let's take a look at the question. Hibernaria is the genus of orchids with dark color and scent flowers, which attract moths for pollination at night. And in the group, H. reticula is an exceptional species with reddish flower which lack a detectable scent. So it was found that no insect visit the flower of H. reticula at night for pollination, while insect species A consume nectar from the flower in the daytime. The flowering dichotomous key can be used to identify the group to which insect species A belongs to. So we have the 3 keys about the wings, antenna, and the wings rest position. So let's take a look at the photograph. Shows the appearance of species A at rest. Write down the sequence which leads to the identification. So this question, it checks us to concept about how to use the dichotomous key to identify the organism. So you can watch this video for the revision. And because we need to write down the sequence which lead to the identification, so we need to use arrow or comma to show the order of the key. You cannot use the space, you cannot use the hyphen to show the order. So let's take a look at the photo. You can see that this species A, it has the wing, so that's why 1A with wing, and then we go to 2, the antenna longer than the head, that's something observable as well. So that's why 2A, 1A arrow, 2A, and then we go to 3. Wings rest at their sides. Wings rest together upright. So what does it mean? Wings rest at their sides. So it should be like this. However, for the species A, when they are at rest, the wings, they rest together upright. Therefore, it should be go to 3B and then it belongs to group S. So you can watch the video to do some revision about the dichotomous key or the classification skills. And then for part B, in this case, H. reticula was evolved from other Hibernaria species, suggests how speciation of the H. reticula could be facilitated by the insect pollinators. So for this question, the main idea is about the speciation and the insect pollinator. That's why we need to recall the mechanism of the speciation, and also we need to recall the source of the genetic variation. Because from the question, it says that H. reticula was evolved from other Hibernaria species. Therefore, they should have common ancestor, right? And then let's take a look at some useful keywords to recall. Prezygotic isolation, that means they are isolated before they can interbreed with each other. Genetic variation, mutation, and the natural selection. Let's go through some critical concepts. How are the common ancestors separate? It's about the isolation. A population separated into small groups are isolated from one another, for example, by the physical barrier, so that they cannot interbreed. So, in the ancestry species of the H. hibernaria, there was no cross-pollination or cross-breeding between the individuals with dark color flower and those with reddish flower because of different insect pollinator. So you can refer to the question. Habenaria, they have the dark color and scent flower, which attract moths for the pollination at night. But for the H. reticula, they attract the species A, looks like a butterfly, collect the nectar, and also do the pollination at daytime. So you can see the timing is much different, right? And then what about the interbreeding? That's something we have already talked about. They cannot interbreed at all. So any criteria for interbreeding or genetic composition, we need to talk about that because they are isolated and they will evolve independently. Therefore, there was no gene flow between these two populations. And after that, they will face natural selection by themselves, separately. So how the species selected by the nature. They have their own mutation, they have their own genetic variation, and after many generations, genetic variation may accumulate to a point where individuals of different isolated groups can no longer interbreed to produce fertile offspring. So you can see that after many generations, mutations, genetic variation accumulate in these two populations such that individuals of these two groups were no longer able to interbreed to produce fertile offspring. You can see how similar is the answer to the textbook. So the main idea is about the isolation mechanism. They are isolated by the insect pollinator behavior. And surely it's also related to their color and the scent as well. And why do they have different color and different scent? Because they have different genetic variation because of the mutation. Therefore, they become two distinct species. The one pollinated by the species A is H. reticula. And you can refer to the question before for the camel, for the flower, for the dogs, for the wolf. We have gone through the mechanism of speciation before. Watch video for the revision. And then let's take a look at part C. To determine if insect species A is attracted to the flower of H. reticula by its appearance or its smell, researcher decide an investigation shown in this diagram. So in this part, I go through treatment 1 and 2 first. Treatment 1, we have the visual setup with the transparent container without host, and the control setup, transparent container without the host and without the plants as well. And for treatment 2, smell setup, non-transparent container with host, and the control setup, non-transparent container with host, and also without the plants. So in each setup, the two setups were in an open area at a distance of 60 centimeters from each other for 1 hour. After that, the position of these two setups was exchanged for another hour. During the 2-hour period, if an individual of species A approached any setup to a distance of less than 10 cm, it was counted as one visit. And the number of visits to each setup is shown in the box in the above diagram. So you can see that for the visual setup, 43 visits, smell setup, 1 visit, and for both of the control setup, there is only 1 visit. For part 1, to ensure that the investigation was a fair test, exchanging the position of the two setups was a necessary step. Explain why. This question, it checks us the importance of the fair test and also the control setup. So the control setup is identical to the experimental setup, except that the factor under investigation is absent. So what is the factor under investigation in this question? Is the plants. So you can see that for the control setup, there is no plants because we want to ensure the result of the experiment is due only to the factor under investigation. That means the presence of the flower of the plants, H. reticula. Secondly, we need to recall the concept of control variables. For the control variable, they are kept constant in the investigation in both setup. Visual setup for the control setup, for example, the size of the container, distance between the container. For example, for the treatment 1, or the distance between the experimental setup and the control setup is 60 centimeters. But for the treatment 2, the distance between them is only 30 centimeters. So it's not fair at all. For the treatment 1, I put them in the open area, which is a park. And then for the treatment 2, I also put them in the open area, but which is a construction site. So you can see that it's not the same. It's not fair at all. So you can identify the CV, right? And what about in this question? We need to exchange the position of these two setups. And it is a necessary step because it is a hidden control variable. We would like to make sure data collected were not affected by the position of two setups. The preference of the species A was regardless of the position of the setups. For example, visual setup is so close to me, so I visit it more frequently. But for the control setup, it's so far away, therefore I do not want to visit it at all. So that's why I exchange the position. Make it from far away to close to you, make it from close to you to far away from you. Thirdly, to cancel the effect of different physical factors of two positions. For example, the temperature, the wind speed, light intensity, so on and so forth. Therefore, you need to give me the named examples. For the last reason is to minimize the effect of the behavioral inertia or habit of the pollinator. For example, the flight time of the species A is just so short. The stamina of this insect is not that high. Therefore, they can just fly for a short distance. Therefore, I exchange the position to try to favor this insect. What we may see is that no matter how far away is the plant, if I want to visit this flower, no matter how far, I will go to visit it. Or for the control setup, there is no flower at all. So no matter how close you put it to the species A, it will not visit to the control setup as well. It's also a simple logic. So you can watch the video before for the revision, especially the comparison, the experimental setup and the control setup, say. And then for part 2, with reference to the aim of the investigation, what conclusion can you draw from the result of treatment 1 and treatment 2 respectively? So for this question, surely is checking us the cyanotic skills, drawing conclusion. And then we need to compare the number of visits of the visual setup and the smell setup with their corresponding control setup, and then use the strict to the point method to draw a conclusion. No need to think too much. You have the result and then draw the conclusion directly. And surely we need to realize the aim of the experiment. So let's take a look at the aim again to determine if insect species A is attracted to the flower of H. reticula by its appearance or its smell. That's why the researcher conduct this experiment. So let's take a look at the treatment 1 first. So in treatment 1, the number of visits to the visual setup is 43 visits, was significantly greater than that of the control setup, only 1 visit. So we recall the result and then we draw the conclusion. This shows that species A was attracted by the appearance of the flower. We have this result to support this idea. And then for the treatment 2, the number of the visits to the smell setup was more or less the same, exactly the same actually, that of the control setup, only 1 visit. And it shows that the species A was not attracted by the smell of the flower. So that's the conclusion drawn by the result directly. No need to think too much. You have the result and you have the conclusion. So you can see that this type of question, drawing conclusion, is also a very popular questions from 2013 to 2020, or even this year 2022. We have MC question, we have long question about drawing conclusion. So these skills you have to be familiar with this. And then for part 3, I want to talk about treatment 3 as well. For the visual and the smell setup, we have the transparent container with holes, the control setup, transparent container with hole but without the plants. And then we have the number of the visits, 48 visits and 2 visits. So let's take a look at the question. When Anne and Ken compare the result of this 3 setup, they have different interpretation. Recall the conclusion in part 2. So we see that all the species A should be attracted by the appearance of the flower but not the smell of the flower. However, they have different interpretation. And thought that the larger number of visits in treatment 3 might reflect a synergistic effect of visual and smell attraction, while Ken thought that it might be due to random variations. To verify Ann's idea, they decide to conduct a further investigation of 2 more treatments for comparison. Question: it checks us the concept how to establish the control setup. Remember that there should be only one difference between 2 setups in each treatment. And then we need to realize the aim of the experiment. We need to verify Ann's idea. Her idea is about the synergistic effect of the visual and smell attraction. Because we can really see that for the treatment 3, for the visual and smell setup, there were 48 visits. And then compare with the visual setup, really more than 43 visits. She may be correct, but can't. Thought that it might be due to random variation. So in order to verify if there is a synergistic effect between 2 stimuli, each stimulus, visual or smell, should be compared with the combined stimuli, visual and smell. So for treatment 4, the control setup should be with the plants, transparent container without the hole. Actually, it is the visual setup. So you can see that I compare the visual and smell setup with the visual setup. And for the treatment 5, I used plants, non-transparent container with holes. Actually, it is the smell setup. So I follow the same logic. Visual and smell setup compare with the smell setup. And the predicted result is that if the visual setup attracts a similar number of visits as that of the visual and smell setup, the presence of the synergistic effect is not supported. Therefore, you have to understand how can we set the control setup, and actually the answer can be reversed. Just one setup with the transparent container without holes, and then the other setup is non-transparent container with holes. One visual setup, one smell setup, and then your answer is correct. And then any possible question variation. So we call the 2022 paper 1B question 5 suggests how to modify this experiment in order to improve the reliability of the investigation. So you can leave your answer in the comment section. Let's see, is your concept clear or not? So let's talk about the curriculum mapping. This question, it starts from the flowering plants, or we call that angiosperm, and then it checks us the concept about the classification and evolution. For the classification, this time we talk about the insect pollinated flower. What about next time we talk about the wind pollinated flower? Any comparison between them? Any advantage or disadvantage of these 2 types of flower? For this question, we also talk about the dichotomous key. So remember that we use the external features, the appearance for the reference to do the identification. And then for the evolution, surely we talk about the speciation, and then we can extend the question about the evolutionary tree and watch the video before for revision. And surely this question asks us about the mechanism of the speciation, isolation, genetic variation, lateral selection, and the survival of the fittest. So you can watch the video before not only the MC but also the long question for revision. And then we combine all the idea to ask about the scientific investigation of the pollination. So for the question, it asks us about the control setup, and then what is the control variables. And in this question, it can ask us something more. For example, we have the hypothesis of N, and then what if N's hypothesis is correct? So what is the prediction? What is the predicted result? And then I will give you the result, and then you can do the comparison and draw the conclusion as well.